So the first part of the area of study is looking at kind of immunity as a whole. So we have our little study design dot points that will appear in the corner. So just refer to them. As I mentioned this, the study design is really, really, really important. I've already discussed how you guys are ultimately the second cohort to use this new study design. Um, I guess it is, I mean, I feel like being at the end of study design is definitely a bit better than being at the start just because you've got past practice exams and things like that. But there was, I would say, more stuff taken out of this study design than was put in. So I think that's a good thing that you guys can sort of work with and use to your advantage. In terms of immunity, not a lot of stuff was changed. I think most of the stuff that's in the study design at the moment was in the study design previously, but again, just some things have been cut out a little bit. Um, but yeah, use the study design to your advantage. I would try and not memorize it word for word, but definitely have an idea of all the dot points that are in each area of study, because this can be really helpful when you get to your short answer questions, especially when you don't know what the question is sort of asking of you. Sometimes you just get to a question and you read it and you just don't know, honestly. And you don't know, what it's trying to ask you. You don't know what it could be referring to. And at that point, speaking from kind of my experience, when I get to a question like that, I always just try to think, I almost like run through the study design in my head and I think, what dot point could this possibly be referring to? What dot point are these, VCAR examiners, what are they trying to kind of nudge me towards, if that sort of makes sense? So I feel like having a good grasp of the study design can be really helpful for that. But I'll talk about that, I guess, a little bit later on. Okay, to start with our actual stuff, we're discussing pathogens. So it's important to understand the distinction between different types, how antigens kind of appear on pathogens and that sort of thing. Definitions. So this is sort of the definition in terms of biological agents that can cause disease, basically. So they impair the normal functioning of the host organism. Obviously, they replicate, and that's how they can kind of spread their disease and how a disease progresses, essentially. And this idea of separating our pathogens between cellular and non-cellular, also very important because VCA will, include questions about this. In terms of definitions, sorry, I feel like I'm putting a lot of tips in before I'm getting to the actual content, but definitions are super important in bio, more so than other subjects, I would say. I remember my teacher always going on about keeping a glossary. And I think it is something that's really important because a lot of the questions in bio are definition based. So particularly, I mean, it can happen in multiple choice as well, but particularly a lot of short answer, they can use basically like a lot of marks are associated with using a definition. And I feel like I didn't really like making a glossary, particularly early on. So I used to kind of like ignore my teacher and be like, oh, whatever, like a glossary, like because I hadn't used a glossary in the past, and I feel like I was very sort of stuck in my ways when I was entering year 10. I realized I didn't really say much about how I experienced bio, but I did bio when I was in year 12. So I did 1 and 2 in year 11. I did 3, 4 in year 12. I got a 45 in bio and I did enjoy it, I would say. I think I enjoyed 3, 4, I would say a lot more than 1, 2. Yeah, and I think I was able to do well in it because I sort of was able to figure out, and I think this is a really important point, how to answer bio questions. I always say to my bio students, it's a completely different thing knowing the bio content and being able to score well, or not even score well, but being able to answer bio questions properly and getting the full mark for the question. Again, talking about how I was always stuck in my ways, I never really did, practice questions and glossaries and that sort of thing. I was very much, do your chapter summaries and then, write your notes and then you answer the question, you get it right. But in bio, I found that wasn't exactly true. I would do my chapter summaries, but then, because I think I would get a pretty good grasp of the content, but then initially I basically wasn't answering the questions properly. I think bio is very specific in the way that examiners want you to answer questions in a particular way, and there's definitely a very particular marking scheme. Again, I would say more so I found it to be different to some of the other sciences where maybe that technique, my old technique that I used to use would work fine. But I think for that reason, having a glossary, doing your practice questions, it's all really important because you can know the content like the back of your hand, but if you don't understand what the question is asking of you and you don't know how to phrase all the knowledge in your head properly, it's not going to work out and you're not going to get full marks. So that is something really, really important to be aware of. Okay. I think I've got enough tips shared now. We'll talk about other things later. But in terms of the actual pathogens that you have to know, bacteria are a really important one. They're probably the most common one that pops up on your exams. So unicellular prokaryotes. So they are cellular, but it's just one cell basically compared to your sort of eukaryotes and your more complex ones. So prokaryotes, basically, they're just very, very simple organisms reproducing independently. So via binary fission, that is a big problem for them. I mean, I guess for us because of them, because of that idea that they can reproduce really, really quickly. So obviously we think about pathogens and bacteria in this context of spreading disease, but bacteria are really helpful. We obviously, we'll talk about our normal flora and stuff and how that can form a bit of a defense system. But yeah, you've got bacteria all over you, your skin, your gut, everywhere, mostly everywhere. But in that certain case, they're not pathogenic because they're not causing disease. In fact, they're doing the opposite. They're being helpful. So when we talk about pathogens, it's in that context of causing disease. Sometimes you might call them opportunistic pathogens. So that's when bacteria that perhaps exist on your body that don't usually cause disease, suddenly they get the opportunity to, and then they do so. So that's just a bit of an example there. In terms of the cell wall, this idea of peptidoglycan, it's not something vital to know, but it's something that is important for certain bacteria, and it can be a good target for a lot of antibiotics. And we classify them based on their shape. So you can see you've got cocci, you've got rods, spirochetes, all these different types. It's not vital for you to know, what's what, and you don't need to know specific examples of bacteria, but just understand that they come in different shapes. Typically, the ones used on a lot of Beaker diagrams and stuff like that are your rods. But yeah, there are different types, so just be aware of that. Be aware of some of the common sort of like characteristics. So you can have like flagella, so this thing that almost like a little bit of a tail. You can have your pili, which sort of stick out. Again, you've got your cell wall, you've got like a capsule sometimes. And so all of these things can be sources of antigens. So we'll get into antigens in a second. But basically just these things that can almost be like little tags on bacteria that our immune cells can recognize. So it's important to be able to recognize, these things like flagella and pili, so you can see it on a diagram and know that you're looking at a bacterium, but also so that you understand that the flagella may be part of an antigen that an antibody is specific for example. But yeah, just get very comfortable with knowing key things about bacteria and knowing what they look like on a diagram as well. Okay, so ways that they sort of work: producing toxins, invading tissues, consuming nutrients. So, you've got your exotoxins, your endotoxins. Not a vital distinction to be aware of, but just the idea of exotoxins being secreted while the bacteria are alive, and obviously the toxins can be harmful to our cells, or the host organism cells. And then your endotoxins are basically when the bacteria bursts, when it dies, the toxins that are inside, as in endo, they leak out, and then they can obviously cause a lot of damage as well. So invading tissues, so we kind of think of bacteria as extracellular pathogens, so they exist sort of outside the cell, and then they can come in and invade tissues. They can release these enzymes, release these other toxins that ultimately break down that tissue and break down the cells in that tissue, and that's obviously going to lead to consequences, symptoms, and ultimately sometimes death of those cells and those tissues. Consuming nutrients is a big thing because basically they just outcompete everything else. So you've got your bacteria and they're consuming nutrients that your cells need. Your cells are losing, they're going to die ultimately. So that's often why in bacterial infections you can get changes in the level of glucose, because of this idea that bacteria are consuming the resources that your cells need. And that's kind of pretty general, inhibiting normal cell functioning as well. Okay, hopefully that all makes sense. Viruses are your kind of second main pathogen that VCAR often use. So definition here, obligate intracellular pathogens. So obligate is the way that because viruses are non-cellular, very important to understand that as well. Non-cellular, please remember that. They can't reproduce by themselves, which is why they are an obligate intracellular pathogen. They rely on being inside a cell and having a host and having something that transcribes and translates in order to replicate. So that's why they're an obligate. They can't sometimes invade a host cell and then sometimes they can just reproduce by themselves. They have to basically hijack the machinery or the replication system in order to replicate and then spread those virus particles to other cells. Okay, so then as we've mentioned, so they will basically infect a cell, transcribe, translate the genetic information that sits inside of their little capsid, and then produce more virions or viral particles. Those will then leave the cell, infect another cell, and that's how the infection spreads. And at the same time, they can go around killing those cells as well. So there are heaps of different viruses. You can be based on their genetic information. So they may have DNA, they may have RNA. Bacteriophages they look like little spiders. You've probably seen it when you looked at CRISPR, but they are very specific to bacteria. They aren't really, they don't really infect human cells, so we don't see them as often in our examples or like in VCAR questions. But it might be good to recognize, especially in that context of CRISPR. And there's other different types of viruses. Again, no virus that you need to know specifically. Some viruses will come up more than others. You probably get a couple questions on COVID, HIV, those sorts of things. But there will never be a really specific question about a certain virus because it's not on study design. You know what I mean? You have to know viruses generally, but they'll never ask what is the exact specific mechanism of an RNA virus or HIV, or describe all of the different antigens of COVID and stuff like that. This is another common thing, a little tip with bio as well. You'll often see things that are unfamiliar. Don't let it psych you out so you kind of get lost and think, oh my gosh, no, I haven't revised this. I didn't look at this specific example of HIV, or I didn't look at this specific example of COVID. Because VCAR often will try to distract you, especially in multiple choice questions. They'll give you this whole chunk of information. They'll talk, all about COVID and this and that and blah, 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 all this random information. And you read it and you'll go, oh my gosh, no, I haven't looked at this before. I've never heard of this before. I'm not going to be able to answer the question. And then basically the hidden question will be are viruses, cellular or non-cellular? But they'll sort of hide it amongst all that information. So then students who aren't aware of what they need to know and what they don't need to know might get frazzled. So again, why the study design is important, because in questions like that, you don't need to worry and go, oh my gosh, I've missed something. Because you'll be able to read the question and understand that, they're just providing some extra context and almost trying to distract you a little bit. Okay, so prions are another type of pathogen that we can see down the bottom there. They are also non-cellular. They are ultimately misformed proteins. So prions, basically, they're misformed proteins. You see them often. The disease may spread from animals. So if you have these really harmful misformed proteins, and let's say you get infected with one, so say you may eat meat that's infected with a prion, right? So you'll eat this and you'll obviously eat the protein, and because it's a misformed protein, it will come into contact with the sort of normal version of the protein in your body, and then it will convert it. It will change that normal protein to this misfolded protein, and it's really harmful. It's kind of affects the nervous system, so you get some of the neurological disorders. So if you had mad cow's disease and stuff like that, that's the kind of sort of things you're thinking about with prions. So they are non-cellular. It's important to remember that viruses are non-cellular and prions are non-cellular, but your viruses are, your little hijacking genetic information sort of thing, and your prions are just misfolded proteins, basically. And so prions are thinking more neurological disorders. Prions, I would say they honestly don't come up very often in exams, whereas viruses, you'll be talking about them every second immunity question. Okay, so here's a nice diagram that sort of summarizes this. You don't need to know the cycle of a virus, but it just may help you visualize it. So you've got your virion, which is just a virus particle that enters the cell. You can see its genetic information held sort of within that the center of that particle, and you can see it's like little proteins, spike proteins on the outside. This is a very typical diagram, like as in this of what a virus will look like. You can expect it to look like that in your VCAR exams. But it basically, yeah, will insert its genetic information into the nucleus. We've got transcription going, and then we'll have translation going. Those proteins that genetic information codes for, which is obviously just to create more virus particles. As you can see here, we've got our spike proteins and stuff like that. Those will be translated, and then you get this new virus particle forming. It ultimately exits the cell, and then this will go on and infect another cell. So that's the idea of how viruses work. Super important thing just to remember that it's non-cellular. Okay, so some of our eukaryotic pathogens. So these are cells that are a little bit more similar to our cells. So a little bit more complex. So we've got our fungi. So they, you think of like your fungal infections ultimately. So, like your tinea, your athlete's foot, that sort of thing. Their cell wall is made of chitin. So you can think again, just like peptidoglycan with your bacteria. The chitin is sort of another target that you could use. But yeah, those are your sort of fungal infections. And then your protozoa. So these are your eukaryotic pathogens, but they're single-celled. So I feel like often we tend to think like eukaryotes are multicellular and prokaryotes are single-cellular. Remember that that's not always the case, such as these protozoa that are eukaryotes, but they're a little bit more simpler. They're still unicellular. So these can sort of be quite parasitic. So like Plasmodium is the cause of malaria. So often we think mosquitoes are the cause of malaria. They're the vector. So just like, in Unit 3 when you discuss plasmids and all that stuff and how your plasmid is the vector for sort of carrying your target gene into your bacterium, your mosquito is basically the vector of carrying this protozoa, this plasmodium, into the human, let's say. So yeah, that's the pathogen we're thinking about. And then worms, so what we've got depicted here, your tapeworms, your liver flukes, that's pretty self-explanatory in terms of what they are. They just look like real worms as well. But they can be obviously a lot bigger, and yeah, that sort of parasitic nature, and where they can just take up the nutrients and absorb all of that at the expense of the host. Okay, so those are the pathogens you need to know. Really important. Again, bacteria and viruses, the main ones you sort of think of. Okay, so in terms of our actual sort of immunology stuff here, so an antigen, very important, again, definition to know. So it is a molecule that is capable of inducing an immune response. So we've got our self and our non-self antigens. So on our pathogens, they will have their own little antigens. If we're thinking of a bacteria, maybe just a little protein on their capsule, or if we're thinking about the virus, like a spike protein, that is the antigen. It's just like a little, I like to think of antigens as name tags. It's just like a little name tag basically identifying what that pathogen is. And if you think of a self-antigen, so like your MHC markers, these are again little name tags that say, hey, like I belong to the body, basically. So your MHC markers are your MHC1 in particular are basically your self-antigens. So they are found on all nucleated cells. So be aware that they aren't on, certain cells like red blood cells and things like that, but they have their own little, like they've got their ABO system. So there's still ways of identifying cells there. But your MHC1 are essentially, your little name tags. And the way that they work is, and I think this is important to know. I remember I didn't know this, this sort of second dot point here. I didn't know it until we were kind of further into the area of study. And I think to me, it helps everything click a little bit more, especially when you think about the difference between cytotoxic T cells and natural killer cells. Knowing what MHC1 actually does, as opposed to just being a self-antigen, for me was really helpful. So just bear that in mind as we talk through them. So with the MHC1, I like to think of MHC1 as basically little windows. I think I might elaborate on this when we get to natural killer cells and stuff like that. But they basically show immune cells. So I like to think of yeah, so cells as houses, your MHC1 as windows, and your immune cells as police. So your immune cells go around and they just basically double check everyone's antigens and make sure that there's no foreign antigens hanging around that they need to be a little bit wary of. So the way that they do that is through these MHC1 markers. So that's why I like to think of them as little windows, because basically the police, so the immune cells, they run around and they look in everybody's windows. So they look at these MHC1, and MHC1 shows what proteins are being made in the cell. So they present peptides derived from cytosolic proteins. So your cytosol, proteins that are hanging around in your cytosol, they take like little bits of that and they put them on their MHC1, and then that shows the immune cells, this is what I'm up to inside of my cell. So that's why I think of them as a little window. So then if something is going a bit dodgy in there, in your MHC1, then the police can realize and then they can sort it out. Yes, that's what I like to think of it as. So obviously, if you're making the proteins that you're meant to be making, that's how MHC1 works, is basically a self-antigen, a self-marker. It tells the immune cells, I am a cell from this body. I'm meant to be here. I'm not doing anything wrong. Your class 2 are a little bit different. They are associated with antigen-presenting cells. So not every single cell has MHC2. Only specific immune cells have it. And these will be used, I was going to say window again, but not really window. These will be used basically just to show when you've got a foreign pathogen hanging around the body. Okay, so we'll show a little picture. So here is this idea of self-antigens and non-self-antigens. So your T cell, which we'll get into in a little second, is a really important immune cell. So they go around and they, can check what's going on. And it's this idea that they have different receptors. If there's a theme for immunity, it's receptors, this idea of things being complementary, matching up, detection, all that sort of thing. A very, very important sort of theme of immunity. So your MHC1 markers, as I mentioned, are your self-antigens. And then your, stuff that might be on a pathogen is your non-self-antigen. So T cells are able to basically detect what belongs in the body and what doesn't. So hopefully that makes sense. Okay, so to get us sort of thinking, so when a virus attacks or infects a human cell, what occurs within the cell? So again, think about your viral particles. Think about what our definition was. So an obligate intracellular pathogen. I was thinking parasite. Yeah, obligate intracellular pathogen. So we're going in a cell and we're basically hijacking that machinery. So we are undergoing transcription, we're undergoing translation, and that cell, so our human cell, will begin to become basically a virus making factory. So MHC1 markers, what they present on the outside of cells. So hopefully you guys are all thinking of the little analogy of a window. Okay, I don't know if it's helpful. It was helpful for me. If you think it's stupid, don't use it, think of something else. But yeah, I just like to think of them as a little window. So they present your little proteins that your cell is making. Again, just a way of showing the immune cells that you're sort of doing the right thing. If you think about it, with a virus, if you're infected with a virus, what you would be making, if you're making viral proteins, you're going to be essentially displaying that on your MHC1. So that's this idea of how the immune system detects cells infected by viruses. Your MHC1, so your windows, they'll be displaying your viral particles. So you've got, a home invasion, right? The virus is a home invader. I guess they come in, they start invading, and your windows are still open, and the police come around, your immune cells come around, and they can see through the window that there is someone invading your home, and then they sort it out from there. So that's the idea of how immune cells detect when cells are infected by viruses via their MHC1 markers. Okay, so looking at our first line of defense, so basically first line of defense, I think it should be used kind of colloquially. according to the study design, it's better to say, your innate and your adaptive immune response as opposed to first, second, third. I think that's something my teacher used to tell me as well. So just be aware of that. Sometimes it's easier to sort of comprehend your first, your second, and your third. But technically, like when you're writing, in short answers, I would never really write first, second, or third. I'm sure depends how lenient your examiner is and all that sort of stuff, but it's always best to use terminology that's listed on the study design. Okay, so you've got your non-specific or your innate immunity and your adaptive or your specific immunity. So your non-specific immunity, and it's important to know differences between these. Your non-specific immunity is basically your one size fits all, works really quickly, sort of like instant, like putting a band-aid, ultimately. So whatever the pathogen is, if it's a virus, if it's a bacteria, if it's a prion, if it's a whatever, the innate immune system is going to act in the same way. Again, one size fits all. There is no memory of prior pathogens. So this idea of memory will make more sense when we talk about adaptive immunity, but it's this idea that it's one size fits all, and the cells that are involved with this, you could be infected with the same bacteria over and over again, but they won't recognize it, is the idea, whereas in adaptive, you do recognize it. And then the level of response being the same for each pathogen of the same organism. Again, that idea, if you're infected by the same bacteria over and over again, when innate immunity kicks in, it'll just kick in. It's one size fits all, exactly the same, copy and paste. Whereas with adaptive immunity, because you have that memory of past infections, if the same organism comes along, that memory kicks in really well and you get a bigger response, and then you get a bigger response, and you get a bigger response every time something, the same organism tries to infect, ultimately. Okay, so in terms of the first line of defense, so this is preventing pathogens from entering the individual, really. A big one is intact skin. You can see it's underlined intact. If you just say skin, you will not get the mark because the skin could be open, the skin could be damaged. Intact skin, it has to be closed. So that's obviously a pretty clear physical barrier. And that's why when it's open, that, skin is sort of useless if there's a big gaping hole in it, because then the bacteria can just jump right in. So mucus and your cilia in the airways. It's also important to know what is a sort of physical and what's a chemical, I believe. I think it's in the other dot point, but your physical, your chemical, and your sort of microbiota, it's important distinctions to make. So your intact skin would be more physical. Your mucus and your cilia can ultimately be both a little bit. Basically because the mucus, I prefer to think about it in a physical sort of sense because I think it's easier to explain in a short answer. Your mucus can ultimately trap those pathogens, and then the cilia just pushes it so it gets sort of chucked, back out or like swallowed and stuff like that. But there are chemicals within the mucus as well that can help to sort of degrade that pathogen. So if you're going to describe it in a chemical aspect, just make sure that you're being specific and be mindful that VCAR may ask you, what are some examples of physical barriers? What are some examples of chemical barriers? And you need to know different ones. So a low pH in the stomach. So this is chemical. Stomach acid is very acidic. So it can kill certain pathogens, but of course there are some bacteria that hang around in the stomach as well. So lysozyme, so these enzymes in your tears and in your saliva, they can help to degrade certain pathogens as well. So your good bacteria, this should be referred to as your sort of your microbiome, your microbiota. So they're found in other places on your body. It says a few exceptions, those are just sterile areas, but it's this idea that this microbiome helps because they compete with your bad bacteria. It's like a little bit of a good versus bad. So when let's say you have a poor microbiome, so I was talking about opportunistic pathogens before. If you take some antibiotics, let's say, they may be pretty general, and what they might do is they may end up killing some of the good flora, or like the good bacteria in your body. And what that means is there's sort of less competition, and so sometimes you can get other opportunistic infections, often like thrush can be an example. So sometimes you have a fungus that sort of sits around your mouth, for example, and sometimes if you take antibiotics or even if you take like Ventolin, like if you take steroids, then you basically kill some of the bacteria around, and then those pathogens that are left, they can become opportunistic and kind of grow. So that's why it's really important to keep a nice balance of the bacteria in your microbiome, for that reason, because the competition is really helpful in making sure that they don't get too crazy, but then also that, your actual foreign pathogens don't invade as well. Okay, so in terms of your second line, so let's think, we've gotten something's gone wrong with our first line. Maybe we have a big cut. Now we've got pathogens that are entering our internal environment. So this is when the second line kicks in. This is your innate immunity. So these are your sort of general leukocytes. Again, this is your sort of one size fits all. So this is a nice little map of your leukocytes. The ones that are blocked out are part of the third line, so your adaptive immunity. So we'll look at those a little bit later. But these are the really important cells for you to be aware of. Be aware, because I feel like sometimes I say it and it might not be captured as well. So this is a good diagram. But you'll often see this thing like a monocyte, and they'll often link it to macrophages. So monocytes are just like an earlier version of macrophages. I think it can be confusing. Sometimes students think it's two different things, but you've got your macrophage, which is a really big like main character in the innate immune system. And then your monocyte is just like the baby version of that, basically. Okay, so this is a really, really important table, or just an important thing to be aware of. Flashcards can be good for this, diagrams can be good for this. Whatever it is, just know the main features of these key innate immune cells. So your macrophages, basically we're thinking about them being really good at phagocytosis. We'll discuss phagocytosis in a second, but it's basically just engulfing pathogens. Engulfing pathogens. As in like phagocytosis, like, like exocytosis and endocytosis. Phagocytosis, like they're just like eating the pathogen basically, and they degrade it and chop it up. Your dendritic cells, I should mention as well, they are an antigen presenting cell, so an APC, so that's why they've got MHC class 2. Your dendritic cells are APCs as well, and they're like the best of the best. So your macrophages are very well known for phagocytosis. Your dendritic cells are very well known for antigen presenting. So that's something to be aware of. Neutrophils, these are not antigen presented, so they do not have MHC class 2. They're the most common type of leukocyte that you see sort of in infections. So there's really, really high numbers of neutrophils. And then they ultimately do this like little kamikaze thing. So they undergo apoptosis after phagocytosis. So with macrophages, when they phagocytose something, they eat it up, cool, they move to the next one, eat it up, move to the next one, eat it up, move to the next one. Neutrophils, kind of like when a bee stings someone and then they die. That's sort of the idea with neutrophils. They will engulf a pathogen, and the way they kill it is that they release all of these like really harmful enzymes and toxins or whatever. So it kills the pathogen, but it ends up killing them in the process. So that's kind of a little characteristic about them. Mast cells, really key. You always associate them with histamine, and they're very involved in your sort of allergic and inflammatory responses. Something to be aware of is that they reside in the connective tissue. So they're not kind of floating throughout the bloodstream. They just sort of sit and release the histamine. Natural killer cells also quite important. These work similarly, not the same, but similarly to cytotoxic T cells. So it's important not to get them confused. And they release these two things you have to know, perforins and granzymes, which you can see here. And those basically induce death in certain cells. So they will detect changes in your MHC class 1. So the way that they do this, remember that innate cells are a one size fits all. So what certain pathogens might do, so some viruses and especially some cancerous cells as well, they know that, the police are coming around looking through windows. So what they do is they shut the window. So the MHC class 1, they down-regulate it. So they just don't have an MHC class 1. This is what the natural killer cell will pick up on. So the natural killer cell, again, one size fits all. It's not checking, is this chickenpox? Is this COVID? Is this salmonella? It doesn't care what it is. It just goes around from house to house, and anybody that has their window closed, as in any cell that doesn't have an MHC class 1, they will release these perforins and granzymes, and that will induce apoptosis, so cell death, and that will kill the cell ultimately. The cytotoxic T cell is a little bit different because it's part of the adaptive, so the specific immune system. So we'll talk about that when we get there. But just remember that natural killer cell, just like all of these cells, they don't care what they're killing. They just know that it's a pathogen. Whereas the adaptive system is a little bit different and more specific. Okay, and then lastly, your eosinophils. These are kind of like the random, like nobody really cares about it. But important in your parasitic infections, and they can undergo phagocytosis as well. Yes. Okay. So we have a lot of like little sidekicks that kind of support these immune cells. So your complement proteins, these are really important. So they basically just help these immune cells. So they can enhance phagocytosis. They can bring immune cells closer to the site of infection. They can actually lyse some bacteria themselves. So they form this thing called a membrane attack complex, and they just kind of like sit on the like plasma membrane or the membrane of the pathogen, and they form a little hole, and then it causes all of the contents to sort of leak out of the pathogen, and then they burst and they die. And then they can help sort of work with antibodies as well, which we'll discuss soon. So cytokines are also quite important. They're signaling molecules, and they're basically just methods of communication. So if a cell wants to talk to another cell, they'll release these cytokines. So cytokines are cytokines, really important. Interferons are also very important. They're a sort of subset of these cytokines, and they're released by cells that are infected with viruses. And what they do is they basically warn the other cells nearby, and they tell them, I'm infected by a virus, watch out. What they also do is they will release, if I'm a cell that's infected by a virus, I'll release this interferon and I'll send it to my friends, right? Telling them be careful. But what I'll also do is I'll send it back to myself. So I'll release it and it'll bind to a receptor on my membrane, and it'll tell me like, slow down with your protein synthesis, like you're infected by a virus, relax a little bit. So that's why interferons are really quite important in viral infections. Okay, this is also extremely important, your inflammatory response. This ties in as well with allergies, which we'll get to. So your inflammatory response is part of your innate immune system. So again, it's this sort of one size fits all idea. So we've got damage occurring to the site. So this is our intact skin. We've now got a hole. That first line is breached. So these damaged cells release chemical signals. So the damaged cells around here will detect that they've been, like the cells in the tissue here will detect that they've been damaged, and that will sort of call your innate immune system into play. Remembering that your innate immune system is very quick. So your macrophages are, ready. They're going, okay, I'm coming to the site of infection, time to help out. Your mast cells, which are these ones here, remember that they sit in the tissue. Unlike these other cells, they're not running through the bloodstream. These ones don't move. They sit in the tissue, and they'll kind of recognize like, oh, something's going on here, and they will release these molecules here, the histamine, and the histamine really gets everything going in this inflammatory response. So we have our main sort of symptoms, so redness, heat, swelling, pain, and loss of function. It's like a thing. They're like your 5 like cornerstones, like major things of your inflammation. And they all kind of come as a result of this sort of histamine and this idea of getting your immune cells into this location. So the redness is because you dilate your blood vessels. So your blood vessels become leakier, they become more permeable, they become wider, so vasodilation, and that just increases the blood flow and allows more immune cells to get to the site quicker. The heat is because of that as well, because your blood is warm. The swelling is because your blood vessels become, as I mentioned, like leakier and more permeable, and this allows immune cells to squeeze through the gaps a little bit more easily. So that idea of them becoming more permeable, means that your immune cells can come through. And so again, this causes a lot more cells in this area, a lot more liquid, fluid in this area, and that's why you get this swelling. Pain you get just because of the damage to the area, and again, all of these cells in this one spot. And loss of function is kind of similar to that as well in terms of all this activity going on here. But yeah, that's what you've got. So your blood cells are leaky and more permeable. So all of these, neutrophils, macrophages, your dendritic cells, they're all kind of coming in to help out. And a lot of phagocytosis will go on. And then basically we'll talk about phagocytosis. They'll engulf these pathogens. And then this will ultimately link us to the third line of defense, so adaptive immunity. But in general, that's what we want to think about. So we can see that the blood is leaking out here because of all our immune cells coming in. So these are our phagocytes. So you can see here, it's just engulfing this little pathogen, and it'll absorb it and just chop it up and kill it, basically. Okay, so we'll work through a little practice question. So which of the following matches a cell correctly with its role in an immune response? So basically working through a process of elimination here. So macrophage stimulates inflammation by secreting interferon. No, because cells that secrete interferon, it's your virally infected cells. Dendritic cell presents fragments of antigens to T helper cells. That sounds about right, because remember that dendritic cells are a really good APC, so an antigen presenting cell. Mast cell engulfs bacteria and debris. No, that would be more so a macrophage sort of thing. A mast cell, it's really key with releasing histamine. Remember that mast cells don't move, so they wouldn't make very good things to engulf bacteria. And your neutrophil secretes antibodies. I know we haven't talked about antibodies, but they're associated with the adaptive immune system, and your neutrophil is part of the innate. So that's why B is correct. Okay, so this process of phagocytosis can be seen in this diagram here. So you've got your bacteria or pathogen, it comes in, it gets absorbed into this little vesicle called a phagosome. So your phagosome then fuses with this other vesicle called a lysosome. And in that lysosome, you've got all of these lysozymes, these enzymes that are involved with degrading things and chopping them up basically. So your phagosome, you've got your little vesicle with your pathogen, you've got your little vesicle with your degrading enzymes, or like digestive enzyme kind of, and then you fuse them into one, which is your phagolysosome. And then the enzymes basically get to work, and you can see that they degrade this bacteria, this pathogen, and then this is kind of just expelled. So it's quite a neat process. So this is what we can see here, phagosome, your lysosome. Step 4, you just combine them together. So that's the process of phagocytosis in general. If you are an antigen presenting cell, what will happen is you're, degrading, you're sort of like digesting this bacterium. You will get part of that, so it's antigens. And what you will do is you will go and present it on your MHC2. So your MHC2 cells, just like your MHC1, will sit on the plasma membrane. So we'll take a little fragment of this, showing your antigen, and we're going to put it on our MHC2 markers, which will just sit around here. We will then go and find our, T cell or whatever, and we will show them this antigen, basically. So that's the idea there. And then the adaptive immune system will kick in. So you can see that antigen presentation is the link between your innate immune system and your adaptive immune system. Perfect. Okay, so looking at our lymphatic system, so this is where we're getting into our sort of adaptive stuff here. So the lymphatic system, basically it transports lymph around the body, and lymph is sort of like a filter, filters your blood and all that sort of stuff, and gets rid of like fluid and all that sort of thing there. So it is like kind of closely linked with the circulatory system, but it's not blood, so it's not pumped by the heart. It just moves based on sort of like valves and your muscles moving and kind of shooting the liquid up. So that's your lymphatic system in general. Your lymphatic organs are important because they're where our cells can sort of reside or grow. So lymphocytes will sit in your lymph nodes. So with this process here of your antigen presentation, when you present it to a lymphocyte, you're going to be moving to a lymph node. So in terms of their names, T cells mature in the thymus, B cells mature in the bone marrow. So all of your lymphocytes start in the bone marrow. Your T cells will move to the thymus, and that's where they'll mature. Your B cells will stay in the bone marrow. That's when they mature. Hence T cells for thymus, B cells for bone marrow. And your spleen is kind of involved in this as well. Okay. So now we're getting into this real link here. So you've got your APC, say you've got a dendritic cell, we've had this inflammatory response. We've got an antigen, we're putting it on our MHC2, and we're now going to go all the way to a lymph node. So it might be in like your neck, maybe in your armpit, wherever. But we go to a lymph node, and this is where we're going to interact with a lot of our lymphocytes. So our T cells, our B cells, all that stuff hanging around there. So once we've got our T cells, again, we're getting higher up here in our hierarchy of immune cells, we need basically a more effective system to kick in. So what your adaptive T cells, or sorry, your adaptive cells in general will do is they are very specific. So that's why we've got an antigen. Before we had, our little one size fits all. We don't care if it's a bacteria, if it's a virus, if it's whatever. Now we've phagocytosed this pathogen, and we are finding its specific antigen. Remember, an antigen is like a name tag. So now we're saying, okay, this is the salmonella bacteria, this is E. coli, this is, strep, whatever it is. And we're giving this name tag, and we're showing this name tag to the T helper cell. And the, macrophage or the dendritic cell is saying, the innate cells, we've tried our best, but we need something more targeted here. That's the sort of idea that we're going with. So you can see that here. You don't need to worry about the co-stimulatory ligand. This is what you're really thinking about. So we've phagocytosed this. We've taken some of this antigen, put it on our MHC class 2, and now we're presenting it to the T helper cell. And this T helper cell is specific to this antigen. So this is another thing as well. We're not presenting this to any random T helper cell that we bump into. We're presenting, let's say this is for like E. coli or something like that. We're presenting this to a T helper cell that is specific for E. coli. So this macrophage will be in a lymph node, and it'll take it to a billion different T cells. It'll take it to the COVID T cell. It'll take it to the chickenpox T cell. It'll take it to the, like tetanus T cell, like whatever. It'll take it to all these different T cells. But remember the whole theme of bio, complementary. We're thinking of receptors. So we're finding a T cell that has a receptor that is complementary to this specific antigen. That is a really, really important thing to understand. Okay, so these are all our main players in our adaptive immunity. So don't really worry about like naive and stuff like that. But you've got your B cell. So again, B cell will have sort of a similar receptor like this. It's to a specific antigen. If it's naive, it means like it hasn't come across it yet. So in our bodies, like in 2018, we had our little B cells. Oh, maybe B cells are a bad one for COVID. But like let's just say, for example, right? It may not be accurate. But we've got our little B cell that's hanging around that's specific to COVID or whatever other infection. That would be naive because it hasn't come across its antigen yet. Then COVID came into our body, and this B cell was saying, woohoo, I found my complementary sort of antigen. So that's the idea of when they become like a mature B cell or an activated B cell. Plasma cells and memory B cells fall under this division of B cells. I think I'll probably explain it in a diagram, but this is a good table to refer to. Plasma cells associate with your antibodies. Memory B cell, you just associate it with being your memory. So they just kind of hang around, and they're good for next infections. Helper T cell, like super important, basically your coordinator of the whole adaptive immune response. They activate B cells, they activate other helper T cells, and they activate your cytotoxic T cells. Your cytotoxic T cells, remember I said, were a little bit similar to your natural killer cells. They also use perforins and granzymes to induce apoptosis in infected cells. But instead of detecting when the windows are down, remember that this is part of the specific immune system. So they detect when the specific antigen is being presented on the MHC1. So they're looking in the window, and they're not only looking in the window and saying, oh, there's an invader. They're looking in the window and saying, this is the invader's first name, their last name, their date of birth, all that stuff. They're being really specific. Okay, so this is a nice diagram that sort of encapsulates this. So this is our link from our innate immunity. So we've got our antigen presenting cell here. We've got our antigen. B cells can also be antigen presenting cells. We just don't really like VCA doesn't really use it in this context very much, but they can be. So whatever the case is, often it's an APC. They present our antigen. This T helper cell will now be activated. And what it'll do is it'll make more T helper cells. It'll, what we call clonal expansion is an important thing to understand for all of these. So remember that all of these have a really specific receptor, right? So there's not a lot of them. Like, let's think about the COVID one, right? Before COVID came along, it's not really being used that much. So it would be useless for you to have like 1,000 of these cells, right? So you may have a couple, but once it's been activated, once this T helper cell specific to COVID, in 2019 or 2020, came into contact with this virus, it obviously needs a bit more help, right? So it will go through this thing called clonal expansion. It'll basically just replicate and replicate and replicate. So now we have a lot of T helper cells specific to this COVID antigen. That will then activate your cytotoxic T cells, which is what we've got here. This will also activate your B cells. And the same thing will happen. Clonal expansion, you'll get heaps and heaps and heaps of these cells. So B cells will go into your plasma cells and your memory B cells. So the role of your plasma cells is just to make antibodies. And again, you've guessed it, your antibodies have those specific receptors to that specific antigen. So these are really good for like bacteria and targeting sort of extracellular pathogens. Your cytotoxic T cells, we've talked about like the windows, they're really good for intracellular pathogens and like your cancers and stuff like that. So that's the idea there. Okay, so summarizing this, so we've got a lymph node, our T helper cell is activated. This is going to say, okay, let me find a B cell that has the exact same receptor. I found it, now you're activated. Now this B cell, it's been switched on, it's going to divide and it's going to create so many more B cells, and they're going to be both memory B cells and your plasma cells. So remember, your plasma cells work to produce your antibodies, and those will help kill your bacteria. Your memory B cells, their role is basically just to hang around. And then once this infection is over, the next time that we get infected with COVID, let's say, we've got a lot of memory B cells hanging around. It's their job to divide into more B cells, basically. Okay, so yeah, plasma B cells, antibodies. That's the idea there. So this is ultimately what we can see in terms of our sort of initial infection. And this is your like antigen presenting cells. You've got your T cells and your B cells being activated. You've got your plasma cells and your B cells, and then your plasma cells producing your antibodies, which look like these little Ys. Okay, I am mindful of time, so I'll keep kind of going through it. So T cell responses are targeted towards our intracellular pathogens. So again, think of our viruses, our cancers, stuff like that. So you've got you're at a lymph node, your APCs come along, T helper cells activated. That activates itself really, and it divides, and it creates your memory T cells and your T helper cells. Memory T cells, exactly the same. I will say VCA always talks about memory B cells. They never really talk about memory T cells, but it's just they have the same job. And then your cytotoxic T cells will obviously proliferate as well. So here is how cytotoxic T cells work. Again, instead of your natural killer cells detecting when MHC1 is not there, what they do is cytotoxic T cells go and they zoom in on your MHC1, and they will see that if there are any basically non-self antigens being presented on the MHC1, and then they will release perforins and granzymes and kill it. So if you've got a virus, your window is open, you're showing your viral particles or peptides on your MHC1, your cytotoxic T cell is going to come along. And remember, I can't emphasize it enough, it's all about being complementary, having the same receptor. So this cytotoxic T cell comes along, and its receptor, whatever the shape is, fits really well over this viral antigen. So this is sitting in your MHC1. This is your cytotoxic T cells receptor. It's going to come along and go, this is the COVID, SARS-CoV-2, like whatever. This is the exact specific antigen that I'm looking for. I'm now going to release perforins and granzymes, and I'm going to like destroy this house. Basically, I'm going to kill this cell and kill the virus in it. So that is the idea there. Okay, so allergens are similar to pathogens in an allergic response, but obviously it's this idea of people with allergies and our response to allergens. It's a hypersensitivity. So, you've got things like pollen, dust, peanuts, compared to, if you think about a virus or a bacteria, they can cause a lot of damage, they can kill you, right? So it's valid that the immune system wants to mount a response against them. With allergens, our immune system is basically very hyperactive, and it sees these pollen, these dust, this peanut, which it isn't a source of a threat for us, but for some reason it sees it as a threat, and then it basically initiates an immune response. So a lot of this stuff is quite similar, but a thing that you have to realize with the allergic response is that you have this sort of priming. So what happens is if we've got someone that's allergic to pollen, so the first time pollen is going to enter their body, and we've got our immune system kicking in. So, the same thing that happened with our clonal expansion, all that sort of stuff will be specific to pollen. And we're going to make antibodies to the pollen, right? Because we think it's dangerous. But something that we do is that with these antibodies, we don't really use it to like kill it, I suppose. What we do is we take these antibodies, so they're a specific form of antibodies, IgE, and we sit them on mast cells. So that's obviously a bit different to what you do, like if it's a bacteria. So the first time your body has ever come into contact with pollen, we mount an immune response, specific antibodies are formed. These antibodies go and sit on our mast cells, and that's that. The person doesn't get sick, nothing happens. The second time pollen comes in, that's when we get this full-blown response. So the second time your pollen comes in, and this is this idea of like sort of priming, your IgE, so your antibodies are sitting on your mast cells and they're just waiting. And so as that pollen comes in, what it does is it binds to that IgE, and that causes a really huge response in your mast cells, and histamine is released, and then you have this big inflammatory response. And that's why people with allergies, have inflammation. They've got they get red, throat closes up, snotty, that sort of thing. So hopefully that makes sense. And that's kind of what I was talking about in terms of hypersensitivity. But yeah, the main thing to realize is that we've got our first exposure, like nothing happens to the outside. You don't have that allergic response. But your mast cells are just covered in this IgE. And then the second time it binds to the IgE, and then it causes that response. Okay, I'm going to probably race through this question. So cells of the immune system have different kinds of structures on the surfaces. So you've got self-antigens, receptors for self-antigens, and then receptors for foreign antigens. So hopefully that makes sense. So you've got a self-antigen. So you can see that they've all got three things. So you've got a self-antigen, you've got a receptor for self-antigens, and you've got a receptor for foreign antigens. So from the information, what are we able to conclude? Again, if you want, you can take like a screenshot of it and have time to think about it yourself and stuff like that. But I will move through it. If you want to go back to it on the slides, then I recommend that you do. The way that you can look at this is if you realize that you've got a self-antigen, a receptor for the self-antigen, and a receptor for a foreign antigen, you can basically figure out which one is which on each of these, because two of them will be complementary because you have a self-antigen and a receptor for a self-antigen. So they must be complementary, right? And then the third one should be a random receptor for foreign antigens. So what the question is asking you is, what is a self-antigen for each of these cells? So if we go to cell P, this fits into that. So this must be the receptor and the self-antigen. So this will be the foreign one. So therefore A can't be right. B, so cell R. So this fits into this. So this must be your self-antigen and your receptor. This is your foreign one. Oh, yeah, that's looking pretty right. Let's check the others. Cell Q. So this fits into this. This must be your self-antigen. This must be your receptor. This is your foreign one, which is not right here. And lastly, this fits into this. So this must be the receptor and the self-antigen. this must be the foreign one, which again is incorrect. So the only one that's right is B. I hope that made sense. Feel free to leave a comment in the chat if that didn't work. I know I went through that quite quickly. But again, take time to go back to it using the slides if you want. Sorry, I just know we have a lot.